Okay, so on to part 2 of aerobic respiration, which remember means how the mitochondria works with breaking down food in the presence of oxygen. We just covered step 1, which is about glycolysis, which, by the way, does not involve oxygen at all, and is probably something that all cells have in common, even our little friends, the prokaryotes. But let's talk about eukaryotes, such as our cells. So let's say there is plenty of glucose in the cell, and the cell needs to break down the glucose to extract the energy from it. After glycolysis, we have made a little bit of ATP. We've also made a molecule called NADPH, which is an electron carrier. And finally, we've made pyruvates or pyruvic acids. Those molecules are going to enter what's called the Krebs cycle. That's kind of the old word. A few times in the past, I have seen it referred to as the citric acid cycle, but lately I've seen people start referring to it as the Krebs cycle again. Discovered, of course, by Dr. Hans Krebs. So this time, instead of being in the cytoplasm, we are actually going to move our pyruvates inside the mitochondria. The carbon dioxide that you are exhaling, this is the time at which it is released. So one of the main waste products of the Krebs cycle is going to be the CO2 that you exhale. So the mitochondria are releasing that CO2. It diffuses back out of the cell into the bloodstream. It circles through the lungs, and hence the exhalation there. We're going to make more ATP through phosphorylation, meaning we're going to take an ADP molecule and add a P to it. Okay. And then also those electrons, which we talked about earlier, they're going to be captured by what are called coenzymes, which are vitamins, essentially. So if you've ever wondered why you need a diet with some vitamins in it, vitamins are important in a lot of chemical reactions in the body, and this being a really important one. So you do not need to know or memorize this diagram. It would always, if it were going to appear on a question, would be labeled for you. You just have to be able to interpret what's going on. So main thing to recognize is your pyruvate is going to enter from glycolysis into what's called the Krebs cycle. But remember, we're inside the mitochondria. There's going to be 2 pyruvate molecules. This is one of them. It's a 3-carbon sugar. The 3 circles are 3 carbon molecules. There's going to be a second pyruvate that's going to have the remaining 3. Because remember that glucose is C6H12O6. So we have 6 carbons to wrestle with. Hence the reason why we'll actually run through the Krebs cycle 2 different times in order to process all of them. Right away, your pyruvate is broken apart. And you're going to begin releasing carbon dioxide molecules. Not all of them at once, but some of them. You've got a molecule called coenzyme A that's going to help rearrange that pyruvate into some other forms of molecules. You don't need to know the details, but we're going to take now that molecule, and it's going to go through a cycle of reactions here. We're going to get rid of even more of the carbons and oxygens. You can kind of see what we're doing here is we're stealing a lot of the hydrogens, just like we were doing in photosynthesis with water and breaking apart the water molecule. We're going to be making NADHs, ATPs, and one more new one here called FADH2. You don't really need to know what that means. It's another electron carrier that's going to help in the process of making a whole bunch of ATP. And remember I said before that NADH is the same molecule as NADPH. It's just when it's used in photosynthesis, we stick the P on there for photosynthesis. NADH is a coenzyme, which means it helps enzymes function. And remember that enzymes are responsible for speeding up chemical reactions in the body, such as breaking down sugars to extract energy from them. It's made from the vitamin called niacin. So if you've ever taken a multivitamin, you'll probably see that one on there. And its job is to help carry some of the burden of the electrons so that you can reach the final step. So the final step is something we actually already talked about in photosynthesis. It's called oxidative phosphorylation. Hopefully you'll remember that anytime you see the name phosphorylation, it means you're going to be building some ATP molecules. We are still inside the mitochondria, but now we're in the very inner membranes. So those folds that we were talking about before, we're going to be working on the insides and the outsides of those folds. We're going to be crossing over. The electrons that we took from the Krebs cycle are going to be carried by our electron carriers to what's called the electron transport chain. And yes, it is not the exact same chain, but it is the same idea of electron transport that we saw in photosynthesis. The goal here to keep in mind is we're still trying to break things down even further to extract and produce ATP energy. So I included this little picture that just talks about the significance and the importance of each one of the parts of the mitochondria and what they do. One thing I just want to mention here is that we're going to be working with protons again. Notice in the Krebs cycle, we didn't immediately talk about what happens to the hydrogens, but very similar to photosynthesis, we're going to be taking the hydrogens from the glucose molecule, separating their electron from their proton, creating a proton gradient, and then allowing something called chemiosmosis to happen where we can generate a whole bunch of ATP. I did want to show you what the electron transport chain looks like here. In photosynthesis, remember it involved photosystem 2, photosystem 1, and some proteins in between, which we called the electron transport chain. Here's the same version for the mitochondria. There are a lot more proteins involved. Once upon a time, I used to know all of them. Fun fact of significance, when we were doing evolution and specifically when we were doing cladograms, we talked about how a lot of genetic information to compare species to species is done using a protein called cytochrome C, because anyone who has a mitochondria produces cytochrome C. So this particularly is where that gene is used. This is where that protein cytochrome C is used in the electron transport chain. So anyway, the electron transport chain is a chain of molecules located in the mitochondria where you move electrons through a series of electron acceptors, meaning I'm going to pass it to you, you're going to pass it to the next one. As we pass it, notice our free energy levels begin to drop, so the energy stored within those electrons begins to drop as well. At the very end of the chain, the final acceptor is an oxygen molecule. It's kind of notated here. What's going to happen is the oxygen is going to accept 2 electrons and combine with a proton to produce water. So if you remember from several slides ago, one of the products of cell respiration is going to be water. This is where water is made. So water is produced as a waste product, as a way to get rid of those electrons. The proteins use the energy released from the electron transport transfers to transport hydrogen protons across the membrane. So the purpose of the transport chain is to move some of the energy around so that we can pump a bunch of hydrogen protons across the membrane. And once you create that, you create a whole lot of hydrogen protons on one side of a membrane and not on another side. And that sets us up really nice for something we talked about in photosynthesis called chemiosmosis. So the story here at the bottom, I found a nice picture that summarizes everything. And it shows you the electron transport chain. And it shows you the very final step down here. Oops, need a pen. Which is the chemiosmosis part of it. How you're going to make a whole bunch of ATP. So over here on this side, it describes to you what happens with the electron transport chain. You see the electrons are going to move through a series of proteins, okay? As they do that, they drop in energy levels, and then they pump the hydrogens all onto one side. Not every single one of them, but most of them. And then we release our electrons to combine with a stray hydrogen proton and an oxygen to form water. So P.S., we talked about why is oxygen significant. Oxygen completes this process. So if there was no oxygen in your cell, you could not continue to pump hydrogens on one side. If that didn't happen, then the next step over here of chemiosmosis wouldn't happen. And if you notice, the big product here is ATP. So when oxygen is not present in the cell, you can't complete the chain, you can't pump the protons, and therefore ATP dries up in the cell and the cell dies. So oxygen, turns out, is really necessary for your survival. How much ATP are we talking about here? From one glucose molecule, estimates range between 32 and 34 molecules of ATP, give or take, depending on other needs in the cell. Not all the ATP is going to get transferred and kept and stored in this process. This molecule here, this protein of ATP synthase, is the exact same one that's found in the membrane of the chloroplast. So it's the same chemiosmosis that happens there. It's just happening in a different part of the cell, or even a different type of cell. Okay, so summarizing what we've learned here, we're going to be making a boatload of ATP from a single glucose molecule. Some of it comes from the NADH, some of it comes from the FADH2. The max output is somewhere around 34 ATP molecules for every 1 glucose. That's a lot. I just want to clarify a couple of terms here. For me as a student, I would always get electron transport and phosphorylation mixed up, and I would always just gloss over them. Like, yeah, I know what that means. I want you to know that they are two separate events, but they are highly involved with each other. Electron transport, as you see here, is basically the moving of those electrons. The purpose is to create that hydrogen-proton gradient. The phosphorylation part, remember to phosphorylate means to build ATP. So this final step here, this is really sloppy. This is the final step, that's the phosphorylation part. Okay, so the idea is to take that gradient. You now have a high concentration of hydrogen protons on one side. You open up the channel, the hydrogens flow through. As they do, it spins the synthase molecule and causes the combination or the combining of your inorganic phosphate with your ADP molecules, and that's the phosphorylation part. So just note, they happen in both. Here's just a little reminder of how of course, they do happen in both. Same story, much less detail here. Notice electron transport is in both of them to pump the protons. The protons then flow through back in the chemiosmosis part. Oh, that was weird. So just note that it happens in both, and the goal in both, by the way, was to make ATP. But in the mitochondria, you just happen to make a lot more because you have a lot more protons you're dealing with. Okay, just some highlights of the recaps. What do we make in each step? In glycolysis, remember we did not make the bulk of our energy, nor did we make our carbon dioxide. We rearranged glucose into pyruvate. We made a little bit of ATP in some of those electron carriers. Krebs cycle, we can extract a little bit more ATP from our pyruvates. We make all that carbon dioxide you are exhaling, that your waste product, that's made here. We make a whole bunch more electron carriers. And then in phosphorylation, we produce that water as another waste product. And then the bulk of our ATP is made in step 3. So if I could give you a multiple choice test, I would definitely ask you which of the 3 steps makes the most ATP, and it would be the phosphorylation step. Okay, just a little picture here summarizing everything with the details. Don't get caught up so much in the pictures. Just kind of understand the overall what's going on in each step, the idea that some of it happens in the mitochondria and some of it doesn't. I do want to talk a little bit about what happens when there is no oxygen present. You don't immediately die. Your body has a little bit of a safety net built in. There is something called anaerobic respiration, and of course meaning not oxygen. So if it happens to be that in the cell there's no oxygen present after glycolysis is finished, a new step would take place, and that's called fermentation. So fermentation and anaerobic are synonyms with each other. Fermentation is the preferred method in a lot of creatures, bacteria, protists, and fungus. We study and know anaerobic respiration in them quite well. In the case of prokaryotes, they don't have any mitochondria, so there is no next step. So basically, bacteria don't need oxygen at all. And in some cases, there are some protists and some fungus who don't need it either. There tends to be 2 examples of fermentation. Animal cells, or I should put that down here. Animal cells, we do what's called lactic acid fermentation, meaning let's say you're running a race and or like walking up the stairs in my case, and you're exerting a lot of energy, you need a lot of energy, you're blowing through a whole bunch of ATP, but you're also running low on oxygen. So you start, huffing and puffing. There's your, homeostasis feedback loop kicking in. But if you prolong that and you do that for a long period of time, like let's say you are, climbing a lot of stairs, some cells will be depleted of oxygen. So what do? They switch over and they do what's called lactic acid fermentation. And lactic acid is like kind of toxic in the cell. It's not going to kill you, but it's definitely something that your cell doesn't want to keep. Your cell is going to stay alive by making the bare minimum of ATP. It's only going to make 2 ATP molecules. You're still going to produce carbon dioxide and water, but you're going to make much less ATP here until you can get more oxygen, meaning take more breaths or slow your oxygen needs. You're going to be very depleted of ATP. And that's why we basically just can't run forever. Like eventually you run out of energy in the cell. If you prolong that and you never get oxygen again, then the cell will die. Other organisms will do what's called alcoholic fermentation. And this is basically how they make alcohol for drinking. How they make beer, how they make wine. You use a microscopic organism, let's say a yeast in the case of beer, or even bacteria. You give them a whole bunch of sugar and you deprive them of oxygen. You seal them up tight in a container. What they'll do is they will break down that sugar and produce carbon dioxide, but they will not make a ton of energy. They'll only make their 2 measly ATP molecules. And then they secrete, instead of lactic acid, they make ethanol. So basically that's how you make booze. Don't do that, of course, but that's essentially how it works. So we discovered that by accident. So for an animal, we don't make ethanol, but sometimes you'll notice the next day your muscles will be very fatigued after you did a really strenuous exercise. That's the buildup of the lactic acid in the body. You have to flush it out with water, basically. And metabolize it and get rid of it through your urine in order to make it go away. So I just wanted you to make sure to be aware that sometimes oxygen isn't always there. For some organism, it doesn't matter, like a bacteria. But for some like us who have high oxygen needs because we have high energy demands, we can't sustain the lactic acid fermentation for very long. But it is a way for us to not drop dead on the spot. And that's a very depressing way to end this, but that is the end. Okay, so that's our final new stuff. I'll have a few things for you to work on in respiration, but then we're going to start reviewing for the AP exam. Let me know if you have any questions. Thank you. Have a good one.